This lecture is on blood part 1. In this lecture, we are going to discuss the composition of blood, its function, and hemostasis, which is the prevention of blood loss. What type of tissue is blood? Remember from AMP1 that there are 4 categories of tissues: epithelial tissue, connective tissue, muscle tissue, and nervous tissue. Into which of these groups does blood fall? Well, if you said connective tissue, you would be right. Remember that connective tissue consists of cells surrounded by a matrix. So, if we took some blood from someone and we put it in a test tube and then a centrifuge and spun it down, the heavier elements would go to the bottom and the lighter elements would go to the top. In the bottom, you would see that erythrocytes, or red blood cells, and they would make up 45% of the whole blood volume. Erythrocytes are part of the cells that make up connective tissue. In the middle, you would see this thin white band called the buffy coat, which is made up of leukocytes, or white blood cells, and platelets. These make up less than 1% of whole blood. The white blood cells, platelets, and erythrocytes are the cells in the connective tissue, and they are called formed elements. Floating at the top is plasma, which is this kind of light yellow color. It makes up 55% of whole blood, and this is the matrix. So again, connective tissue, blood is connective tissue. It consists of cells, the formed elements, surrounded by the matrix, plasma. It just happens that the plasma is fluid. Now, one common test that's done in hospitals is something called a hematocrit. And a hematocrit is a measure of the percent of formed elements in blood. It is really mainly looking at red blood cells, or erythrocytes. And the way that this is done is pretty similar. So the blood is spun down, but they're just really measuring the column of erythrocytes. And this is an indirect measure of the oxygen carrying capacity of blood. The more red blood cells, the more oxygen. Normal values for men are 46 plus or minus 5%. So this means 46 plus 5% would be 51. 46 minus 5% would be 41. So the range, normal range is 41 to 51 for men. For women, normal values are 42 plus or minus 5%. So the question is, why do men have more red blood cells than women? Now remember, this is a percentage, so you can't say it's because they're bigger. The main reason actually is because men make more testosterone, a hormone that increases the production of erythrocytes by increasing a hormone called erythropoietin. Women make testosterone too, but they make a lot less of it. Another reason is that women lose a little bit of blood every month through the menstrual cycle, and this actually adds up to result in them having a little bit lower hematocrit. Let's look at some of the physical characteristics of blood. Blood is viscous. It is 5 times more viscous than water. Now remember, viscous means the thickness of blood, or the thickness of a fluid. Its color ranges from scarlet, and I put a little example of what scarlet looks like here, and scarlet would be an oxygen-rich blood, to a dark red or a maroon color found in oxygen-poor blood. So blood is never blue. Some of the models that you might have looked at will show the veins as being blue. So they color the veins blue and the arteries red. If you've dissected a cat or a fetal pig, they'll often inject blue latex into the veins and red latex into the arteries. However, blood is never blue. The reason they do this is to help you identify them. Blood may look blue if you look at the veins, for example, on your wrist, but this is just really a trick of refraction. So basically, veins are fairly superficial. And what happens is that the skin is reflecting the blue at you before the hemoglobin can absorb it. So hemoglobin normally absorbs blue, reflects red, and that's why blood looks red. But arteries are fairly deep, veins are fairly shallow, And so the skin reflects the blue before the hemoglobin can absorb it, and that's why you see blue. The bottom line is blood isn't blue. The pH of blood ranges from 7.35 to 7.45. And this is a very narrow range that the pH must stay within in order to be in homeostasis. If you go significantly below this, not even significantly below, maybe to 6.7, people could die. And the reason is that at a lower pH, the hemoglobin is not able to bind the oxygen. So the red blood cells are shuttling through the body, but they're not carrying any oxygen. Blood is about 8% of the body weight, and the average volume of blood in males is about 5 liters, and about females, it is about 4 liters, and that is because of the difference in size. So what are some of the functions of blood? Well, we can put them in a couple of categories. The first category is transport. Blood transports things. For example, it transports oxygen and CO2 and other gases. It transports nutrients. Remember that hormones travel through the blood. In addition, waste products of cells are released in the blood and travel that way, and something called electrolytes, which are really just ions, such as sodium, calcium, it's a hydrogen ion, hydroxyl ion. These are just a couple of examples. Now, another function of blood is regulation. Blood regulates your body temperature. The main source of heat in the body is skeletal muscle. So remember that when skeletal muscles use ATP, when they hydrolyze it, a lot of heat is released. What happens is that blood absorbs the heat released from active skeletal muscles. And then it distributes it throughout the body. And in this way, blood regulates your heat. Now, another way it does it is that when you are overheated, there are blood vessels on the surface of your skin which dilate. That means they enlarge, and as they do, the blood comes to the surface, cools off, and then dives back down. So this is a way of cooling off your body. When you are cold, the blood vessels on the surface of the body constrict. And by doing this, they keep the blood away from the surface, which is cold, and in the core, which is warm. So this is one way that blood regulates your body temperature. Another thing it regulates is pH. Remember the pH of blood has to stay within a very narrow range between 7.35 and 7.45. This is done by buffers. Now remember that buffers, if you remember this from biology, are chemicals which either absorb or release hydrogen ions. And hydrogen ions are what make something basic. So let's take an example here. Let's say up here that this blood is too basic. So the pH might be, I don't know, 7.6, too basic. And remember, just as a reminder, pH goes from 14 to 0. Where 7 is considered neutral, 0 is extremely acidic. So you're getting more acidic as you go down, more basic as you go up. So let's say it's too basic. And here is the buffer. I'm going to call it B. So too basic, you do have hydrogen ions out here, but let's just say you have an awful lot of hydroxyl ions, and there are hydrogen ions also carried by the buffer. If it's too basic, then the buffer is going to release hydrogen ions. And as it does that, the pH goes down. Let's say the blood is too acidic. In this case, we'll say the pH is 7.1. Okay, that's too low. It needs to be 7.35 at least. So here's the buffer again, carrying some hydrogen ions. But now we've got an awful lot of these outside in the blood. In this case, the buffer is going to absorb some of these hydrogen ions, remove them from the blood, and that's going to cause the pH to go up. So again, one of the things that blood does is regulate the pH. Another thing that blood does is it regulates blood volume. Now by this I mean it keeps the blood in the blood vessels. You don't want the blood to move into the tissues when it's not meant to happen. So when you have a massive movement of blood from blood vessels to tissue, that drops the blood volume and can result in stroke. So we're not talking about tissue exchange with the capillaries here. We're talking about a big movement, and you don't want that to happen. The way that works is by having the proper osmotic pressure with electrolytes. So blood maintains the proper osmotic pressure, and it does that with electrolytes. So just a quick review here. Osmosis is the diffusion of water. Remember that? Another way to look at it is it's the ability of a solution to attract water. So when you look at a blood vessel, inside the blood vessel are a lot of things, but you've got, of course, you've got ions here, or electrolytes. And you also have those ions and electrolytes in the tissue. Remember that the tissue has fluid in it. So there's fluid here in the tissue, and there's fluid inside the blood vessel, blood. So the ability to attract water is osmosis. You don't want a net flow out. So you don't want a net flow of water out of the blood vessel. That could result in shock. And this proper balancing of electrolytes controls the movement of water. So blood volume within the blood vessel is regulated by the concentrations of electrolytes within the blood. In addition, blood volume is regulated by blood because it controls hemostasis, which, remember, is the loss of blood. Now, the last function of blood is protection. Blood actually prevents blood loss by initiating clot formation when a blood vessel is broken, and it also prevents against infections because white blood cells are also part of your immune system, and they travel in the blood. Let's look now at the components of blood, and we said there's plasma, which is the matrix, and they're the formed elements, which are the cells. So let's start with the plasma. Plasma mostly consists of water, and it is a clear yellow solution. It distributes heat through the body, and it also distributes all of these other substances. So proteins, such as albumins, globulins, and clotting proteins, the wastes from cells travel in the plasma, nutrients, electrolytes, and gases. Let's take a closer look at plasma proteins. Now, most plasma proteins are produced in the liver, and there are three major categories. The first are called albumins. Albumins are major plasma proteins. They act as a carrier for some molecules through the blood. For example, lipophilic compounds, which cannot dissolve in blood, basically they don't dissolve in water, have to be carried through the blood by another compound, and albumins do that. An example of a lipophilic compound would be steroid hormones. Steroid hormones do not dissolve in blood, and so they are shuttled through the blood by albumins. There are also several toxic lipophilic compounds found in the environment, like endocrine disruptors, which mimic the effects of estrogen. Albumins will bind to these lipophilic toxic compounds and diminish their toxicity. Albumins also act as a buffer, maintaining the pH of blood, and they're important in maintaining the proper osmotic pressure, as we already talked about. The next group is called globulins. Globulins consist of three major groups: alpha, remember the Greek symbol alpha, beta globulins, which transport proteins, fat-soluble vitamins, for example, vitamin A, vitamin E would be an example, and hormones that don't dissolve easily in the blood, such as estrogen. These are steroid hormones, testosterone, thyroid hormone, and corticosteroids. The other thing that's transported by globulins are apolipoproteins. Now, you might remember from the cardiovascular system that we know some lipoproteins. Remember HDL and LDL, high density lipoprotein and low density lipoproteins. These lipoproteins, remember, bind lipids. In the case of HDL, they are going to bind lipids from the tunica intima and remove them. LDL is going to drop off lipids onto the surface of the tunica intima. Well, once these cells bind to a lipid, they are no longer an apolipoprotein. So A means not. So this basically means what these are before they have lipids bound. These are all also transported by globulins. Lastly, we have gamma globulins. There's the Greek letter for gamma. These are antibodies, which are part of your immune system. The last group of plasma proteins are called clotting proteins, and these make up about 4% of plasma proteins, and they're involved in clotting. And we're going to talk about these later. So that is the plasma, and these are the things that are traveling in the plasma. Now let's take a look at formed elements. Formed elements, remember, are the living cells. They are made in the bone marrow, and they only survive a few days. They also do not undergo mitosis. There are three groups. First, we have the erythrocytes, which are also called red blood cells. We have the leukocytes, also called white blood cells, and we have platelets. Now, the only one of these that actually has a nucleus is leukocytes, and we're going to see that as we go through this. So let's say we're going to look at a human blood smear, and you have to do this as part of your lab on the differential white blood cell count. But if you looked at a human blood smear, you would see that most of the cells here are erythrocytes. Here in the middle, we see a leukocyte. This happens to be a monocyte, bigger than the red blood cell. And these little dots here is not actually dirt. These are platelets. So erythrocytes are the most abundant type of cell we're seeing in blood. So let's take a close look at erythrocytes. These are biconcave discs. That means they cave in on the top and they cave in on the bottom. They cave in on two sides. So the middle is very thin, and that's why it appears kind of white here, very thin. These are very small cells, and they are anucleate. Now remember, if you see A before a word, it means not. So they have no nucleus and pretty much no organelles. In fact, they are almost completely full of a protein called hemoglobin, abbreviated HB. Hemoglobin is a protein that transports oxygen. Men have about 4.5 to 6.3 million red blood cells per cubic millimeter. Cubic millimeters, centimeters are often used in medicine. In science, they tend to use microliter. So 1 cubic millimeter is basically a microliter. In women have about 4.2 to 5.5 million red blood cells per cubic millimeter. And again, the reason for this is that men make more testosterone, which promotes more red blood cell production. So let's take a look at the structure of hemoglobin. And you'll see here that there are actually 4 polypeptide chains. So we have this one down below, that's alpha. They call this alpha 1, alpha 2, beta 1, beta 2. So we have 4 polypeptide chains. And then within the center of each polypeptide chain is this green disc. If we look a little closer, we'll see that this is actually heme. This is heme. Now, heme is a cyclical structure, but you'll notice in the middle is iron. This is where oxygen binds, right here. So each iron binds 1 molecule of oxygen reversibly. Each iron, which is in the middle of each heme, binds 1 oxygen. So, how many molecules of oxygen does 1 hemoglobin molecule bind? How many molecules of oxygen does 1 hemoglobin molecule bind? Well, if you said 4, you would be right, because of course 1 oxygen binds to each heme, and we've got 1, 2, 3, 4 hemes in a hemoglobin. So 1 hemoglobin molecule binds 4 molecules of oxygen. Now the next question is, 1 red blood cell has 250 million molecules of hemoglobin. That's 2.5 times 10 to the 8th molecules of hemoglobin. So how many molecules of oxygen does 1 red blood cell bind? So think about that. So we know that 1 hemoglobin molecule binds 1 molecule of oxygen. 1 iron binds 1 molecule of oxygen. 1 hemoglobin binds 4 molecules of oxygen. So how many molecules of oxygen does 1 red blood cell bind, which has 250 million molecules of hemoglobin? Well, you just multiply it by 4 and you end up with 1 times 10 to the 9th, or 1 billion molecules of oxygen. That is a lot of oxygen. 1 red blood cell binds 1 billion molecules of oxygen. Do we need 1 billion molecules of oxygen? Probably not. In fact, now I'm going to tell you that in the human body there is 2.5 trillion red blood cells. So how many molecules of oxygen can the blood in your body bind? Too many to count is a good answer, or 2.5 times 10 to the 30, which means add 30 zeros after 2.5. The point here is that hemoglobin is very efficient at binding oxygen. And so our bodies are able to carry more oxygen than we need. Remember that structure determines function. This is a theme throughout our course. So what is it about the red blood cell that makes it so perfect at carrying oxygen? Well, first of all, it's got that biconcave shape. And remember, anything that has indentations in it that isn't a circle is going to have a greater surface area. And the greater the surface area, the more oxygen, the more room for gas exchange. The other thing is that there's pretty much nothing inside the red blood cell. There's no nucleus, there's no organelle, so about 98% of a red blood cell is hemoglobin. Okay, so there's plenty of room in a red blood cell for hemoglobin, which binds oxygen. Now, red blood cells are living structures, so they need ATP. However, they don't use oxygen to make it. So remember, the most efficient way to make ATP is aerobic respiration. If you were going to use aerobic respiration, you'd have to use some of the oxygen you're transporting to the tissues. But instead, if we look at a red blood cell, we would see no mitochondria, which is where aerobic respiration occurs. So red blood cells produce ATP anaerobically through anaerobic respiration, which is perfect because then they're not using their payload that they're supposed to be delivering to tissues. So again, the structure of a red blood cell is perfectly developed to support its function, and that's why it does what it does. Now, just like structure determines function, if there's something wrong with the structure, there's going to be a problem with the function. So I'm going to mention this one disease, sickle cell anemia, which is an inherited disease. It's caused by an abnormal hemoglobin, and they call that abnormal hemoglobin HbS for HB sickle. If you have 2 copies of HbS, one from the mom, one from the dad, you're homozygous and you have the condition called sickle cell anemia. So what's going on with this? Well, here is one hemoglobin molecule, and they're showing you the amino acid sequence. So we're just showing you the first 7 amino acids, and this is a normal red blood cell. But here you'll notice in the sickle cell, there's one amino acid that's off, and that's number 6, valine. So instead of glutamine, you've got a valine. Now, a lot of times these type of changes don't make any difference in the function, but in this case it does. So what happens is that when people are under low oxygen conditions, for example, let's say someone was exercising or they were in a cave with low amounts of oxygen, then in people with sickle cell anemia, the beta chains link together and form these stiff rods. And those rods distort the shape of the cell. And it can distort into a sickle shape, but there are many other shapes it can take. It can look like this, kind of like a sword. There's all kinds of shapes it can take. But in any case, the distortion of the shape means that it no longer can bind hemoglobin, no longer can bind oxygen, I'm sorry. So there are some red blood cells that are carrying oxygen, but most of them are distorted and they're not carrying any oxygen. And this can cause very, very dangerous problems for the person. They can have a heart attack, they can have a stroke. The edges of these distorted cells are spiky, they're sharp, and they can get caught in a blood vessel and cause a jam, a log jam, or a clot. So it's a very serious disease. Now, if you've only got one copy of the sickling gene, so you only have one HbS, but the other hemoglobin is normal, then you're said to have sickle cell trait. People with sickle cell trait often do not have symptoms. But when people who have sickle cell trait are infected with malaria, it somehow gives them resistance to the malarial parasite and allows them to resist malaria. And this is an example of coevolution. Okay, so a couple of things about hemoglobin. There are different names for it depending on what's bound to it. When oxygen is bound to hemoglobin, they call it oxyhemoglobin. When oxygen is not bound to hemoglobin, they call it deoxyhemoglobin. Carbon dioxide can bind to hemoglobin also, but when it's bound, they call it carbaminohemoglobin. Oops, that's an O. Now, the reason they don't call it carboxyhemoglobin is because it doesn't bind to the iron. So remember, oxygen binds to the iron, that's why they call it oxy. But carbon dioxide doesn't bind to the iron because, of course, you wouldn't want it competing on iron with oxygen for the binding site. Instead, it binds to amino acids on the polypeptide chain. And so the name amino is giving you a clue that the carbon dioxide does not bind to the heme. Now, there is one other chemical, carbon monoxide, CO. Carbon dioxide is CO2. And carbon monoxide is produced by the incomplete combustion of fossil fuels. So things like propane or oil, gas, natural gas, will produce carbon monoxide. In most homes, however, in all homes, this carbon monoxide, which is an odorless, colorless gas, is vented safely to the outside of the home. However, sometimes there are leaks in the ductwork, and the carbon monoxide can build up in the house. In this case, the carbon monoxide binds preferentially to hemoglobin. That means if you have the same amount of oxygen as you do carbon dioxide in the air, the hemoglobin is going to bind the carbon dioxide. And it's going to bind to the iron. In this case, they call hemoglobin carboxyhemoglobin. Now this is very dangerous because instead of having oxygen, you've got carbon monoxide, which does not meet the needs of the cell. And you end up with decreased oxygen in the circulating blood. How much of a problem is this? Well, at 0.1% carbon monoxide in the air, 50% of a person's hemoglobin would be oxyhemoglobin, and 50% would be carboxyhemoglobin. This would result in death in 4 hours. Some of the symptoms of carbon monoxide poisoning, and this happens every winter in New England, there's a tragedy where it happens, are flu-like symptoms. So people feel like they have the flu and they go to bed and then they don't wake up. Also cherry red skin. If someone seems to have carbon monoxide poisoning, they should be removed to the fresh air. And carbon monoxide should dissociate from hemoglobin and be replaced with oxygen. If they've been exposed for too long, then sometimes they have to take them to a hospital where barometric chambers will increase the pressure on their bodies, and also pure oxygen gets piped in, and this forces the carbon monoxide off the hemoglobin molecule where it's replaced with oxygen. Okay, the next thing we're going to talk about is the production of blood cells, and the term for that is hematopoiesis. Okay, so there are a lot of vowels here. O-I-E-S. Hematopoiesis means blood cell formation. So this is how all blood cells form. Red blood cells, white blood cells, and platelets. Hematopoiesis occurs in the red bone marrow. So in an adult, there's a fair amount of red marrow in the proximal epiphysis of the humerus, or the proximal epiphysis of the femur, also in the hip bone, also in the sternum. But usually if someone is giving a bone marrow transplant, they're going to avoid the sternum and go for the bigger bones. In this area, you will find what's called a hemocytoblast. So in the red marrow is a hemocytoblast or hemopoietic stem cell. They both mean the same thing. These are stem cells that give rise to all types of blood cells. So we're going to start by looking at how red blood cells form. Production of erythrocytes, it's called erythropoiesis. And we're going to start over here. Now this whole process takes about 15 days. And here we are in the red marrow, and we're looking at a stem cell, a hemocytoblast. So on the surface of this stem cell are receptors. Okay, these are supposed to be receptors, and they might be receptors for different hormones or different growth factors or other chemicals. What happens is that these receptors bind to certain growth factors or hormones, and once they do, that pushes the cell to become committed. Now, a committed cell just means its pathway is determined. That means it is now going to be an erythrocyte. It is not going to be a leukocyte or a platelet. Committed. Its pathway is determined. And then it starts doing things that it needs to do to become a red blood cell. So you'll start to see ribosome synthesis occur in the cell. Now remember, ribosomes are necessary for protein synthesis. So this is one of the components. So that's a clue when you start to see lots of ribosomes made that proteins are starting to be made. And of course, hemoglobin is a protein. You'll also see the cells start to accumulate iron. So it's starting to make proteins, which is the globin part of the molecule, and it's starting to accumulate iron, which forms the heme part. It puts them together, and now it starts accumulating hemoglobin. As it does, the nucleus gets smaller and smaller because it's being squished by all the hemoglobin. At some point, the nucleus is ejected, and the cell collapses inward. In the space the nucleus used to be, it collapses. At this point, we have what's called a reticular site. Now, in the reticular site, there are still some rough endoplasmic reticulum There's still a few here, some ribosomes, but pretty much this reticulocyte is good to go. So it is going to be released to the blood where it will circulate for maybe 1 or 2 days before it matures into a red blood cell, erythrocyte. So again, this whole process takes about 15 days. Physicians will often measure the amounts of reticulocytes in an adult because healthy adults should have 1 to 2% of their blood consisting of reticulocytes. This indicates a healthy level of red blood cell formation. Now, what regulates this production? Well, the number of erythrocytes, remember, has to stay within this narrow range. So that means you want to produce them and destroy them at a rate where they stay steady at a certain range. Erythropoiesis is mainly controlled by hormones, and we're going to talk about that hormone in a minute, but it's also dependent on having the adequate supplies of the building blocks. So you need to make sure that you've got enough iron available, amino acids to make the polypeptide chains. You also need carbohydrates and the B vitamins such as B6, B12, and folic acid. Okay, so let's look at this hormonal control of erythropoiesis. And here we see this kind of seesaw here. Now the parameter we're looking at is normal oxygen levels. So what regulates erythropoiesis, or I should say the stimulus for producing more red blood cells, is not actually the number of red blood cells. It is the amount of oxygen in your blood. So the trigger for the release of this hormone, for this whole process here, is the oxygen levels in the blood. If the oxygen levels in the blood are too low to meet the tissue demands, then you're out of homeostasis. And as this blood travels through the kidney is going to release a hormone called erythropoietin. Now, the kidney is not an endocrine gland, but it does have endocrine tissue. The erythropoietin is going to travel through the blood to the red marrow, and here we have a femur. And it's going to stimulate, or I would say speed up, the production of erythropoiesis. Okay, so just a minor point here. It does not initiate the production of new erythrocytes, but it speeds up the process of erythropoiesis that's already ongoing. And you end up with more red blood cells, which means you increase your oxygen carrying capacity, and now you're back in homeostasis. Okay, so that's this process. Once erythropoietin is released, it can enhance erythropoiesis incredibly. So it can increase red blood cell production from 2 to 30 million red blood cells in a second. So with the release of erythropoietin, you can jump from the production of 2 million red blood cells a second to 30 million. And one thing I want to mention is that some people have taken note of this process. And of course, having a higher oxygen level in your blood can give you an edge if you're an athlete. If you've got more oxygen, you can make more ATP, which means that you can enhance your performance in terms of energy, speed, and endurance. So some athletes have illegally done something called blood doping, which is when they remove some of their blood, they centrifuge down just the red blood cells, and then they put them in the refrigerator and wait till just before an athletic event. Then they re-inject the red blood cells in them, and now they have more red blood cells, so they have more oxygen carrying capacity, which gives them an edge in terms of their performance. Of course, this is illegal, and it's also dangerous because when you have more red blood cells, you increase your viscosity of blood, which means you could have a stroke. The other thing some people have done is they've just injected erythropoietin, which actually brings about the same result, but this is also illegal. Okay, so now that we know about how red blood cells are formed, let's look at their entire life cycle. Remember that red blood cells only live about 120 days, and the production of red blood cells is about 2 to 3 million red blood cells per second. So we're producing quite a bit. And of course, the stimulation for production comes from the amount of oxygen in the blood. So up here we have the release of erythropoietin traveling to red marrow and speeding up the process, and we've got the new red blood cells, and they're going to circulate in our circulation for about 120 days. Now, towards the end of this time, they become old and damaged, and they are constantly being filtered through your liver and through your spleen. If we take a look at the liver, once the blood cells become damaged, they are broken down by macrophages into their building blocks, which is basically hemoglobin, because remember, red blood cells are pretty much hemoglobin. Your body is a great recycler. It will recycle anything it can to use again. And so the globin is broken down to amino acids, which are released to the blood to be taken up by other tissues. The heme contains iron. Now, iron by itself, the iron ion, and by that I mean Fe+2, can also be Fe+3. This particular ion is very toxic when it's by itself. So what happens is the body binds iron, the iron ion, with another compound, and it's stored in the liver in ferritin or hemosiderin. So this is a compound, because you can use iron again. And then when you need it, it's transferred and released as it's needed for erythropoiesis. Now the rest of the heme molecule is really waste, and that's something, it forms something called bilirubin. Bilirubin is released through the bile to your intestines, and in the intestines are bacteria that will break it down to something called stercobilin. Bilirubin is yellow, stercobilin is brown, and then it's excreted in your feces. So of course, some of that color for the feces is really broken down components of red blood cells.